So the next is the Baltimore system. We will continue with the taxonomy. First, we have studied about the ICTV system, which started from the order, and then it family, subfamily, and then genus, and then species of the viruses. Next is the Baltimore system. Baltimore system was started from the David Baltimore in 1971, and it has classified the viruses into different classes, which maybe we have studied this one. The group 1 is maybe DNA viruses, and then RNA viruses. DNA viruses positive strand or negative strand, then RNA viruses again the positive or negative strands, and then the double-stranded RNA viruses, single-stranded DNA viruses. So different kind of viruses we will study in this. All these kind of viruses will be replicated by different ways of the replication. It all depends on the expression of the nucleic acid, whether the nucleic acid have been the DNA or RNA, and then the DNA is the single-stranded or double-stranded, or the RNA is the single-stranded or double-stranded or stranded, whether they are the negative sense or positive sense of DNA and RNA, segmented or not, and the mode of replication. As far as the Baltimore system of the classification is concerned, we can divide the viruses into 7 different classes. First of all is the group 1, which contains the double-stranded DNA viruses. The double-stranded DNA viruses means that they are just normal the DNA viruses, which are the double-stranded, just normal. For example, in example, we will consider that adenoviruses are the most importantly double-stranded DNA viruses, then the herpesvirus, also the DNA virus. Then there is the group 2, which are single-stranded DNA viruses. This is the only ability of the viruses that they have the single-stranded DNA viruses. In this, the example is concerned the parvoviruses, which are the single-stranded but DNA viruses. Then the group 3, it has the double-stranded RNA viruses. For example, the reoviruses are very important feature, and they have the double-stranded RNA viruses. Group 4 is the positive sense single-stranded RNA viruses. The most common of them are the polioviruses and picornaviruses, which are prevalent in the human beings, and these are the positive sense single-stranded and RNA viruses. Then the group 5 is the negative sense single-stranded RNA viruses, and the example is the arthovirus, which are very common, and they are the single-stranded and negative sense. Then the single-stranded RNA viruses, just like the retroviruses. The 7th class are the hyperdNA viruses, which are double-stranded and DNA viruses, which is the example is the hyperdNA viruses. So from the Baltimore classes or classification, we have these 7 classes of the viruses, which are all dependent on the nucleic acids, whether DNA or RNA or single-stranded or double-stranded, or the positive sense or negative sense, etc. Then the viral classification we can see from this similar way from this table. First of all, the DNA viruses. DNA viruses will be again classified into 2 viruses, double-stranded or single-stranded DNA viruses. On the left side, we have taken the double-stranded and then single-stranded. Then single-stranded viruses will be again whether they are the enveloped or enveloped viruses, just like parvoviruses. However, there are now enveloped viruses, for example, single-stranded, then the double-stranded, then will be enveloped and unenveloped. We have given the examples of some viruses here for the enveloped viruses, and similarly the double-stranded unenveloped viruses, adenovirus, papillomaviruses, and poliomyeliviruses are the unenveloped single double-stranded DNA viruses. Then the other classification, the major classification, is the RNA viruses. RNA viruses will be again in the double-stranded or single-stranded, positive sense RNA or negative sense RNA. First of all is the positive sense RNA, then again the enveloped or unenveloped viruses. Caliciviruses or coronaviruses are unenveloped and enveloped viruses examples. Then the negative stranded RNA, this will be only contain the enveloped viruses, which will be Rna viruses or bunny viruses, filoviruses are the examples of RNA negative stranded viruses. Then the positive sense or negative sense RNA viruses, it will be again classified into the presence of the capsid. It contains the double capsid, and the example is the reoviruses. Then the positive RNA via the DNA, just like the double-stranded enveloped viruses. Example of this virus is the retroviruses.